Welcome back to Concepts of Biology. We'll continue to read Chapter 4, Chapter Summary. Section 4.1, Energy and Metabolism. Cells perform the functions of life through various chemical reactions. A cell's metabolism refers to the combination of chemical reactions that take place within it. Catabolic reactions break down complex chemicals into simpler ones and are associated with energy release. Anabolic processes build complex molecules out of simpler ones and require energy. In studying energy, the term system refers to the matter and environment involved in energy transfers. Entropy is a measure of the disorder of a system. The physical laws that describe the transfer of energy are the laws of thermodynamics. The first law states that the total amount of energy in the universe is constant. The second law of thermodynamics states that every energy transfer involves some loss of energy in an unusable form, such as heat energy. Energy comes in different forms: kinetic, potential, and free. The change in free energy of a reaction can be negative, meaning releases energy, exergonic, or positive, consumes energy, endergonic. All reactions require an initial input of energy to proceed, called the activation energy. Enzymes are chemical catalysts that speed up chemical reactions by lowering their activation energy. Enzymes have an active site with a unique chemical environment that fits particular chemical reactants for that enzyme, called substrates. Enzymes and substrates are thought to bind according to an induced fit model. Enzyme action is regulated to conserve resources and respond optimally to the environment. Hi, if you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. All right, let's get back to reading. Section 4.2, Glycolysis. ATP functions as the energy currency for cells. It allows cells to store energy briefly and to transport it within itself to support endergonic chemical reactions. The structure of ATP is that of an RNA nucleotide with 3 phosphate groups attached. As ATP is used for energy, a phosphate group is detached and ADP is produced. Energy derived from glucose catabolism is used to recharge ADP into ATP. Glycolysis is the first pathway used in the breakdown of glucose to extract energy. Because it is used by nearly all organisms on Earth, it must have evolved early in the history of life. Glycolysis consists of 2 parts. The first part prepares the 6-carbon ring of glucose for separation into 2 3-carbon sugars. Energy from ATP is invested into the molecule during this step to energize the separation. The second half of glycolysis extracts ATP and high-energy electrons from hydrogen atoms and attaches them to NAD positive. 2 ATP molecules are invested in the first half, and 4 ATP molecules are formed during the second half. This produces a net gain of 2 ATP molecules per molecule of glucose for the cell. Section 4.3, Citric acid cycle and oxidative phosphorylation. The citric acid cycle is a series of chemical reactions that removes high-energy electrons and uses them in the electron transport chain to generate ATP. 1 molecule of ATP, or an equivalent, is produced per each turn of the cycle. The electron transport chain is the portion of aerobic respiration that uses free oxygen as the final electron acceptor for the electrons removed from the intermediate compounds in glucose catabolism. The electrons are passed through a series of chemical reactions with a small amount of free energy used at 3 points to transport hydrogen ions across the membrane. This contributes to the gradient used in chemiosmosis. As the electrons are passed from NADH or FADH2 down the electron transport chain, they lose energy. The products of the electron transport chain are water and ATP. A number of intermediate compounds can be diverted into the anabolism or other biochemical molecules such as nucleic acids, non-essential amino acids, sugars, and lipids. These same molecules, except for nucleic acids, can serve as energy sources for the glucose pathway. Section 4.4. Fermentation. If NADH cannot be metabolized through aerobic respiration, another electron acceptor is used. Most organisms will use some form of fermentation to accomplish the regeneration of NAD+, ensuring the continuation of glycolysis. The regeneration of NAD+ in fermentation is not accompanied by ATP production. Therefore, the potential for NADH to produce ATP using an electron transport chain is not utilized. Section 4.5. Connections to other metabolic pathways. The breakdown and synthesis of carbohydrates, proteins, and lipids connect with the pathways of glucose catabolism. The carbohydrates that can also feed into glucose catabolism include galactose, fructose, and glycogen. These connect with glycolysis. The amino acids from proteins connect with glucose catabolism through pyruvate, acetyl-CoA, and components of the citric acid cycle. Cholesterol synthesis starts with acetyl-CoA, and the components of triglycerides are picked up by acetyl-CoA and enter the citric acid cycle. This is the end of Chapter 4. In our next section, we will read Chapter 5, Photosynthesis.